Good morning students and welcome to video lecture on Biomolecules 2. Proteins are the fundamental structural material of animal bodies. Protein in the form of enzyme play prime role in all the physiological reaction. On page number 307 there are some questions. Can you recall what is the product of reaction of acetic acid with ammonia? The answer of this question is ammonium acetate. Second question is write the structural formula of N-methyl acetamide. What is the name of the functional group in this compound? This CONH2 amide group. And what is the nitrogenous nutrients in human diet? The proteins. So, protein perform many essential physiological functions including catalyzing biochemical reactions. Alpha amino acid proteins on complete hydrolysis give rise to mixture of alpha amino acid, and alpha amino acids are carboxylic acid having amino group bonded to the alpha carbon atom, that is carbon next to carboxylic group. Alpha carbon in all alpha amino acid is chiral. It is found that alpha carbon atom in alpha amino acid obtained by hydrolysis of protein has L configuration. Now next is types of proteins. Depending upon the molecular shape, proteins are classified into two types, globular protein and fibrous protein. Molecules of globular protein have spherical shape. This shape results from coiling around the polypeptide chain of protein. Globular proteins are usually soluble in water. For example, insulin, egg albumin, serum albumin, etc. Fibrous protein. Molecules of fibrous protein have elongated rod-like shape. This shape result in holding of polypeptide chain of protein parallel to each other. Hydrogen bond and disulfide bonds are responsible for this shape. Fibrous proteins are insoluble in water. For example, keratin, myosin. Proteins are large biomolecules or micromolecule consisting of one or more long chain. Most animal sources of proteins are meat, egg, beans, vegetables, nuts, often lack one or more essential amino acids. Amino acids are the structural unit that make up proteins. They join together to form short polymer chain called as peptide or long chain called either polypeptide or proteins. These polymers are linear and unbranched with each amino acid within the chain attached to two neighboring. A molecule containing amino acids and carboxylic acids that are separated by carbon are called as alpha carbon atom. See the structure of amino acid structure, amino group, carboxylic group and side chain. Then nutritional classification. Essential amino acid, semi-essential amino acid, in summary of essential and semi-essential amino acids and non-essential amino acid. Zeta ion. Although alpha amino acids are commonly zeta ion in the unionized form, they are more properly written in zeta ion. Both the NH and COH group in the amino acid undergoes ionization in water. Physiological pH 7.4. Zeta ion form both positive and negative charge. Overall neutral. Amphoteric amino acid is protonated carbonyl group is deprotonated and soluble in polar solvent due to ionic character. Then primary structure of protein. The primary structure of protein. Primary structure of protein is the sequence of constituent amino acids residues linked by peptide bond. Any change in the sequence of amino acid residue results in different protein. Primary structure of proteins is represented by writing the 3 letter symbols of amino acid residue as per the sequence of concerned protein. The symbols are separated by dashes according to the convention. N-terminal amino acid residue as written at left side and C-terminal amino acid residue at the right side. The primary structure of protein. Then denaturation of protein. High temperature, acid, base and even agitation can disturb the non-covalent interactions responsible for specific shape of protein. This is denaturation of protein. Denaturation is a process by which molecular shape of protein changes without breaking the amide or peptide bond that form the primary structure. Denaturation results in disturbing the secondary, tertiary or quaternary structure of protein. This causes change in properties of protein and biological activity is often lost. So, in this video lecture, we will discuss proteins, classification, denaturation of protein and amino acid classification.